Hi, my name is Ariel Madrigal. I'm a PhD student from McGill University, and today I have the pleasure to talk to you about our project, which is called a Unified Model for Bayesian Integration and Interpretation of Single-Cell RNA-seq Data. One of the problems that exists in the single-cell analysis field is a problem known as integration. What we have here is that we have unwanted sources of technical variation present within our dataset. When we perform, for example, dimensional reduction embedding of our data, what we might observe is that we have clusters that instead of representing the cell type of origin, they represent the batch of origin. In order to solve this problem, different computational methods have been developed over the recent years with the idea of removing these unwanted sources of technical variation. However, more complex scenarios might arise. For example, we may have the same problem as before, but now with the additional consideration that we have two biological conditions of interest. Now, the problem is that we want again to remove unwanted sources of technical variation, while also learning something about the differences between our two biological conditions. In order to tackle this problem, we have developed GLBIO. GLBIO stands for Gene Expression, Decomposition, and Integration. What GLBIO does is that based on a set of vectors, it creates a reference frame, which establishes a manifold. In this manifold, different cell types of populations may lie in. When we analyze a given sample, we might be able to quantify the differences that would occur to the reference frame in order to match the transcriptome of this sample. When we look now at multiple samples, we might realize that the transformations of the reference frame might be different across samples. Finally, we can connect these distortions to the reference frame to sample-level variables. GLBIO can also be used to model the manifold as a function of gene-level variables. And what we're doing here is that we're using prior information about pathways, gene regulatory networks, and cell type signatures in order to guide the creation of the reference frame. GLBIO can also work with different types of data distributions. It can work with either with normalized data, raw count data, or it can model the log-of ratio of two entities, which is useful for the analysis of splicing and stability. The first thing that we wanted to do was to evaluate GLBIO's ability to capture sample-to-sample variability. We look at this data set, which is a collection of different immune cell types profiled across multiple single-cell RNA-seq technologies. And then what we did is look at the differences to the reference frame for each individual sample, and then use those differences and apply a UMAP to them. So in this plot, what we're seeing is each dot represents one sample. And in this sample level analysis, what we're able to recover is variability associated with the two donors while removing variability associated with the different technologies. Now we can do this analysis, but also at the single cell level. So in these plots, each dot represents one single cell. We're able to recover the structure of the different cell types, remove variability associated with the different technologies, and we can also quantify this performance with this overall score and compare it to other integration methods. Our results show that GLBIO stands out as the top performer method. Having established that we can capture sample-to-sample variability, the next thing that we wanted to do was to model sample variables in GLBIO. To do this, one way to visualize the differences that would occur between samples is through these transcriptomic vector fields. Now, the concept here is that we have our reference frame defined by our set vectors, and what we wanted to do now is to measure the change that will occur to the reference frame given a change in a sample level variable. We want to now to apply this concept by looking at this COVID-19 dataset, which is a collection of different immune cell types from 40 donors, but now with 3 disease groups. So we have controlled, mild, and severe COVID-19 cases. We can generate an embedding that recapitulates the cell type structure, but now we can learn something about the differences between severe and controlled cases. So we can visualize these differences with our transcriptomic vector field. So this vector field represents the changes that will occur to a given cell state, to a given cell, going from the control to the severe COVID-19 condition. So the vector field is a way of representing these changes, and it's a cluster-free approach, but we can also calculate the mean per cell type and compare it to a pseudobulk approach. Our result shows that our mean vector field are highly concordant with the gene expression changes from the pseudobulk. GLBIO can also be used to model the manifold as a function of in-level variables. And the concept here is that we are projecting the activity of a given transcription factor into an embedding. Then we can calculate the gradient of this transcription factor activity, exemplified by this arrow that goes across the diagonal. Then we can compare this gradient vector field against other transcriptomic vector field to see if they are pointing in the same direction. Now we wanted to look at this concept, again using the COVID-19 dataset, and identify transcription factors whose gradient correlates with the vector field of severe COVID-19. We identified this transcription factor called SPI1. We can visualize its gradient of the vector of the transcription factor activity, and then we can compare it to the vector field of the severe COVID-19. What we observed is that for the monocyte population, there seems to be a cell state enriched with high gene expression changes that occur in severe COVID-19, and that the activity of SPI1 correlates with these gene expression changes. Finally, GLBIO can also be used for the analysis of ratio data, in which we are modeling the log-odds ratio between the reads that would support an exon inclusion event and the reads that would support an exon exclusion event. We apply this concept to this mouse neurogenesis dataset, where we are integrating the exon inclusion and exclusion counts. We're able to remove technical variability associated with the study while recovering the different cell type structure of the data and identifying novel exon inclusion events that are cell type specific. So in summary, GLBIO decomposes the effect of different sources of sample-to-sample variability. GLBIO learns cluster-free gene expression changes along a continuum of cell states and reconstructs COVID-19 associated vector fields. GLBIO identifies transcription factors whose gradient correlates with COVID-19 associated vector fields, and GLBIO allows the study of alternative splicing landscapes. With this, I want to conclude, and thank you so much for your attention.